Hi guys, Shweta is welcoming you at the Pharmacy Crash Course Division in the Knowledge Club online. We completed the anatomy part of the heart in previous 4 episodes. From today, I came with the physiology part of the heart. Today's topic is the physiology of the cardiac muscle. The anatomy part of the heart will be used in this physiology part. So, revise it thoroughly. Don't skip today's lecture. If you skip the lecture, you can miss a valuable tips or tricks for your exam. So, stay tuned up to the end. Cardiac muscle tissue is only found in the heart. Highly coordinated contractions of cardiac muscle pump blood into the vessels of the circulatory system. Similar to skeletal muscle, cardiac muscle is striated and organized into sarcomeres, possessing the same banding organization as skeletal muscle. However, cardiac muscle fibers are shorter than skeletal muscle fibers and usually contain only one nucleus, which is located in the central region of the cell. Cardiac muscle fibers also possesses many mitochondria and myoglobin, as ATP is produced primarily through aerobic metabolism. Cardiac muscle fibers cells also are extensively branched and are connected to one another. Cardiac muscle is similar to skeletal muscle in that it is striated and that the sarcomere is the contractile unit, with contraction being achieved by the relationship between calcium, troponins and the myofilaments. This article will consider the structure of cardiac muscle. We learned at anatomy part of heart, the interior of the cardiac chambers is lined by the endocardium, which is formed of a simple layer of cells and is continuous with the vascular endothelium. The muscle layer is situated between the endocardium and the epicardium. It is also called myocardium, right? If you missed the lecture, please visit our channel or you will find link at the description part of this video. Coming to the point, the individual cells of myocardium are called cardiac myocytes. The cardiac myocytes are interconnected to form a functional syncytium. These connections are two types, mechanical and electrical. So, starting with the mechanical connections. The mechanical connections are the intercalated discs and the desmosomes. The intercalated disc makes strong end-to-end connections between the fibers. These connections fall on the Z lines of the consecutive myocytes and provide attachment to the thin filaments. The desmosomes are present at points near the intercalated disc in between the adjacent fibers. Then, what are the electrical connections? The electrical connections between the myocytes are formed by gap junctions. The gap junctions are present where the adjacent fibers come in close contact, side to side. Gap junctions are also present in the intercalated discs. These gap junctions are formed of proteins called connexons, which provide low impedance electrical paths from one cell to another. The basic organization of thick and thin filament in cardiac muscle cells is comparable with that in skeletal muscle. Electron microscopy reveals repeating light and dark bands that represent I bands and A bands respectively. Thus, cardiac muscle is classified as a striated muscle. The Z line transacts the I band and represents the point of attachment of the thin filaments. The region between two adjacent Z lines represents the sarcomere, which is the contractile unit of the muscle cell. The thin filaments are composed of actin, tropomyosin and troponin and extend into the A band. The A band is composed of thick filaments along with some overlap of thin filaments. The thick filaments are composed of myosin and extend from the center of the sarcomere towards the Z lines. Myosin filaments are formed by a tail-to-tail association of myosin molecules in the center of the sarcomere, followed by a head-to-tail association as the thick filament extends towards the Z line. Thus, the myosin filament is polarized and poised for pulling the actin filaments towards the center of the sarcomere. A cross-section view of the sarcomere near the end of the A band shows that each thick filament is surrounded by 6 thin filaments, and each thin filament receives cross-bridge attachments from 3 thick filaments. This complex array of thick and thin filaments is characteristic of both cardiac and skeletal muscle and helps stabilize the filaments during muscle contraction. Several proteins may contribute to the organization of the thick and thin filaments, including meromyosin and C protein in the center of the sarcomere, which appear to serve as a scaffold for organization of the thick filaments. Similarly, nebulin extends along the length of the actin filament and may serve as a scaffold for the thin filament. The actin filament is anchored to the Z line by alpha-actinin, whereas the protein tropomodulin resides at the end of the actin filament and regulates the length of the thin filament. These proteins are present in both cardiac and skeletal muscle cells. The thick filaments are tethered to the Z lines by a large elastic protein called titin. Although titin was postulated to tether myosin to the Z lines and thus prevent overstretching of the sarcomere, there is evidence indicating that titin may participate in cell signaling, perhaps by acting as a stretch sensor and thus modulating protein synthesis in response to stress. Such signaling by titin has been observed in both cardiac and skeletal muscle cells. Moreover, genetic defects in titin result in atrophy of both cardiac and skeletal muscle cells and may contribute to both cardiac dysfunction and skeletal muscle dystrophies, termed titinopathies. Titin is also thought to contribute to the ability of cardiac muscle to increase force upon stretch. Within cardiac muscle cells, myofibrils are surrounded by the sarcoplasmic reticulum, SR, an internal network of membranes. This is similar to the SR in skeletal muscle, except that the SR in the heart is less dense and not as well developed. Terminal regions of the SR above the T-tubule or lie just below the sarcolemma or both and play a key role in the elevation of the intracellular Ca2 plus or calcium 2 plus during an action potential. The mechanism by which an action potential initiates release of calcium ion in the heart, however, differs significantly from that in skeletal muscle. The heart contains an abundance of mitochondria. Up to 30% of the volume of the heart is occupied by these organelles. The high density of mitochondria provides the heart with great oxidative capacity, more so than is typical in skeletal muscle. The sarcolemma of cardiac muscle also contains invaginations, T-tubules, comparable to those seen in skeletal muscle. In cardiac muscle, however, T-tubules are positioned at the Z lines, whereas in mammalian skeletal cells, T-tubules are positioned at the end of the I bands. In cardiac muscle, the connection between the T tubules and the SR are fewer than and not as well developed as those in the skeletal muscle. The myocardium is composed of two types of cells. Number 1, working myocardial cells or myocardial contractile cells, and number 2, myocardial conducting cells or the cells which form the junctional tissues of the heart. The working myocardial cells or myocardial contractile cells form the walls of the cardiac chambers. They contract and generate force. These are long fibers and attached to the skeleton of the heart as well as encircles the cavities. So their contraction reduces the size of the cavities through reduction of both the transverse and longitudinal diameters. They constitute the bulk, 99% of the cells in the atria and ventricles. Contractile cells conduct impulses and are responsible for contractions that pump blood through the body. The myocardial conducting cells form the conduction system of the heart. The junctional tissue is made up of modified myocardial cells specialized in producing and conducting electrical activities. It consists of the SA node, AV node, internodal tract, bundle of His and Purkinje fibers. These cells form the pacemaker of the heart, that is, the places where electrical impulse is produced automatically. Cardiac muscle is myogenic, creates its own action potentials, but is regulated by the pacemaker cells, the sinoatrial node and atrioventricular node. These cells spontaneously depolarize the trigger a cardiac action potential. However, the cells of the bundle of His and the Purkinje cells are also capable of spontaneous depolarization. However, it should be noted that Purkinje fibers are not cardiac muscle cells. They are larger cells with far fewer myofilaments and extensive gap junctions, as well as lots of glycogen. They conduct action potentials much faster, allowing for synchronous contraction of the ventricles. Except for Purkinje cells, they are generally much smaller than the contractile cells. So, what is the anatomy of cardiac conduction? We know now the 5 structures comprises the cardiac conduction system. They are sinoatrial node or SA node, the pacemaker. Number 2, atrioventricular node, AV node. Number 3, atrioventricular bundle, AV bundle, and left and right bundle branches. Number 5, Purkinje fibers. So the sinoatrial node or SA node, a small knot of cardiac muscle like tissue. The pacemaker cells look like cardiac muscle cells, but they have lost the ability to contract. Located on the back wall of the right atrium, near where the superior vena cava enters the heart. The SA node cells initiates the electrical impulse that generates the heartbeat. Atrioventricular node, a similar small mass of tissue located in the right atrium but near the septum that divides the right and the left atria from the ventricles. Its function is to relay the impulses it receives from the SA node to the next part of the conduction system. Atrioventricular bundle, AV bundle, a bundle of fibers that extend from the AV node into the interventricular septum, which divides the heart into right and left sides. The AV bundle transmits cardiac impulses. Left and right bundle branches where the septum widens. The AV bundle splits into the right and left bundle branches, each extending down under the endocardium and then up along the outside of the ventricles. These bundles carry cardiac impulses. The last one is Purkinje fibers. At the end of the epibundle branches are the Purkinje fibers, which deliver the impulse to the myocardial cells, causing the ventricles to contract. The word node is used in many different contexts and not only in anatomy. In general, it describes a local connecting point of component parts, literal and figurative. In cardiac anatomy, a node is a specialized type of tissue that looks like muscle and generates electrical impulses like nervous tissue. At last, some important points of myocardial cell structure. Number 1. The sarcomere. Sarcomere is the contractile unit of the myocardial cell. Is similar to the contractile unit in skeletal muscle. Runs from Z line to Z line. Contains thick filament myosin and the thin filament actin, troponin, tropomyosin. As a skeletal muscle, shortening occurs according to a sliding filament model, which states that thin filaments slide along adjacent thick filaments by forming and breaking cross bridges between actin and myosin. Number 2. Intercalated discs. Intercalated discs occur at the end of the cells, maintain cell-to-cell cohesion. Number 3. Gap junctions are present at the intercalated discs. Are low resistant paths between cells that allow for rapid electrical spread of action potentials. Account for the observation that the heart behaves as an electrical syncytium. Number 4. Mitochondria are more numerous in cardiac muscle than in skeletal muscle. Number 5. T-tubule are continuous with the cell membrane, invaginate the cells at the Z lines and carry action potentials into the cell interior. Are well developed in the ventricles but poorly developed in the atria. Form ducts with the sarcoplasmic reticulum. Number 6. Sarcoplasmic reticulum are small diameter tubules in close proximity to the contractile elements. Are the site of storage and release of Ca2 plus for excitation contraction coupling. So this is the overall structure of the cardiac muscle. Revise it thoroughly because this cardiac muscle chapter will be used for the next episode for studying the action potential of the heart. Write me at the comment box how much you enjoyed today's episode, and also you can write the questions for any confusion. Before ending today's episode, if you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. And after subscribing our channel, please click on the bell button. After clicking the bell button, you will get our updates regularly. One announcement here. If you or your family members have interest in Indian history, then you can follow our another session, the Itihash Katha, where you can know the true stories of the Indian history, where the narrator name is Mr. Shohito Banerjee. I am coming with another episode very shortly. Till then, goodbye.